Good afternoon, I'm Meng, a postdoc associate in plant breeding and genetics at Cornell University, working with Dr. Mike Gore. The title of my presentation is Elucidating the Gene Regulatory Landscape of Natural Variation for Leaf Cuticular Conductance in Maize. The cuticle is a hydrophobic layer of cutin and waxes, synthesized by plant epidermal cells. Plant cuticle consists of a matrix of cutin with cuticular wax embedded within or deposited onto its surface. Cuticular conductance, or GC, is the rate of water evaporation from the leaf cuticle, which accounts for almost all the non-stomata water loss. To investigate permeability of leaf cuticle along its developmental stages, our collaborators performed a stunning experiment and a dehydration experiment using developing maize leaves. They showed that the last mature section or the bottom section of the leaf were more permeable to TBO, a water-soluble dye, and had a higher rate of water loss, indicating that matured leaf cuticles could reduce plant non-stomata water loss compared to immature cuticles. Many studies for maize leaf cuticles were performed on its juvenile leaves, which have more epicuticular waxes and have very different wax composition from adult leaves. In this study, we focused on cuticular conductance for maize adult leaf because they are more related with plant reproduction stages. Our primary goal is to identify candidate genes that regulate leaf GC through integrating genomic and transcriptomic data. The second goal is to validate the association between GC and our strongest candidate gene using maize nested near isogenic lines, or NILS. Cuticular conductance and transcript abundance were profiled for a subset of 310 inbred lines in the Wisconsin diversity panel. The entire panel, with B73 as checks, were planted in two environments in University of California, San Diego, 2018. GC was evaluated for the mature primary ear leaf at silking. The harvested leaves were kept in dark before and throughout phenotyping to make sure stomata were closed. Leaves were weighed every 45 minutes for 5 times, and GC was calculated as leaf weight loss per hour adjusted by leaf surface area. For 3' RNA sequencing analysis, we dissected the bottom 10 to 30% of the total leaf length for a developing adult leaf as it poked out of the hole. Due to the destructive RNA sequencing sampling methods, GC phenotyping was performed on different plants from the same plot. To analyze the data, I calculated fast linear unbiased predictors, or blobs, for GC to combine phenotype from two environments. To get high-density SNP markers for GWAS, the genotypes at over 14 million SNP loci were imputed based on high-quality GBS SNPs in the Wisconsin diversity panel using BEAGLE. After filtering for imputation accuracy, minor allele frequency, and inbred coefficient, about 9 million SNP markers were retained and used for GWAS. Transcript abundance from two environments were combined by calculating blobs for each gene model. The probabilistic estimates of expression residuals, or PEER, were applied to blobs to account for inferred confounders that influence expression variation. PEER residuals of blobs were used for TWAS. To increase the statistical power of our association analysis, p-values of GWAS and TWAS were integrated in the Fisher's combined test. Here is a summary of GWAS and TWAS results. Briefly, we identified 17 plausible candidate genes involved in the cuticle biosynthesis, cell wall modification, intracellular trafficking, and the regulation of cuticle development, as indicated by these color dots. Fisher's combined test integrated GWAS and TWAS results and identified additional 5 plausible candidate genes, as highlighted by red arrows, indicating that Fisher's combined test could increase statistical power in candidate gene identification. Among these candidate genes, ISTL1 was the only plausible candidate identified by all 3 methods. Therefore, it was considered as the strongest causal gene for GC. Here is a regional Manhattan plot that combined GWAS and TWAS results for the 1 megabase region surrounding ISTL1. Vertical lines represent minus log 10 p-values for GWAS and dots for TWAS. We see that ISTL1, whose position is indicated by a vertical dashed line, is tightly linked to a GWAS peak, and its TWAS signal is moderately strong, as indicated by a red diamond. This gene encodes an increased salt tolerance-1-LIKE1 protein involved in the formation of multivesicular bodies. The multivesicular body is a late endosomal compartment where plasma memory-derived proteins are degraded after internalization via endocytosis. In rhabdopsis, ISTL1 and another protein work together to control the plasma memory localization of protein, including ABCG9, a transporter of trafficking component in the tapetum of rhabdopsis. Moreover, others of ISTL1 mutant represent reduction in the accumulation of wax component in the alkane and sterol groups. Thus, it can be speculated that ISTL1 ortholog may facilitate the localization of plasma memory transporter required to export cuticle component in maize epidermal cells and regulate leaf cuticular conductance. To further investigate the involvement of ISTL1 in GC, we selected a set of 12 maize NILS that harbor the ISTL1 region from 8 donor parents with B73 background. These NILS, donor parents, and B73 as checks were planted in 2 environments at UCSD in 2020 for GC evaluation. Best linear unbiased estimators, or BLUES, for GC were calculated to allow for the comparison of GC between B73 and NILS plus their donor parents. In addition, a haplotype-based analysis was performed in both the diversity panel and NILS in order to validate the association between GC and ISTL1 genomic region. This is a scatter plot showing BLUES for B73 NILS and their donor parents. Dot types represent the genetic sources of ISTL1 introgression. We compared GC between B73 and the other experimental lines, and the p-values were adjusted using the Donna's method. 4 of the donor parents, KI11, NC350, OH43, and TX303, showed significantly higher GC than B73. Most importantly, compared to B73, 2 NILS with TCI8 introgressions and 1 NIL with OH43 introgression showed significantly reduced GC, as highlighted in red boxes. To take one step further, we also want to know if the association between GC and the ISTL1 genomic region can be validated among NILS. To do such comparison, we first constructed haplotype blocks and performed haplotype-based association analysis in the Wisconsin diversity panel. And this regional Manhattan plot shows results for haplotype-based association analysis in the Wisconsin diversity panel. Each of the horizontal lines represents a haplotype block, and the length of the line corresponds to the size of a block. Blue means a haplotype block is polymorphic among NILS, and red represents a monomorphic block among NILS. The green narrow region represents the physical position of ISTL1. We focused on a -+200 kb window from ISTL1, as highlighted in yellow background, which is the same window we use for candidate gene selection for GWAS. In this window, we selected the top 10 most associated blocks, as highlighted by a red box, to test their association with GC among NILS. Unfortunately, haplotype block 95, the most associated block in the diversity panel, was not polymorphic among the NILS, which reduced the statistical power and partially explained why we didn't see much significant difference in GC between B73 and NILS due to the introgressions. Of the 10 mostly associated haplotype blocks, 3 haplotype blocks were significantly associated with GC among NILS. As these 3 blocks are in linkage disequilibrium and showed similar effect on GC, I will only focus on block 82 in next slide. In the Wisconsin diversity panel, we estimated haplotype effect for 10 haplotypes in block 82. Among them, haplotypes 1, 5, and 7 were found among NILS. Comparing haplotype effects for haplotypes 1, 5, and 7 from the diversity panel and NILS, we observed strong correlation with correlation coefficient of 0.92. Although the association were estimated from 3 haplotypes, it indicated that the association between GC and ISTL1 genomic region detected in the Wisconsin diversity panel could be validated in an independent population. In conclusion, GWAS, TWAS, and Fisher's combined tests identified candidate genes involved in cuticle biosynthesis, cell wall modification, intracellular trafficking, and regulation of cuticle development of leaf cuticular conductance. Among all these plausible candidate genes, ISTL1 is the strongest candidate causal gene that regulates maize leaf cuticular conductance. TZI8 introgression for the ISTL1 region can significantly reduce maize leaf cuticular conductance, and the genetic effect of ISTL1 region identified in the Wisconsin diversity panel was verified in a haplotype-based association analysis using NILS. With that, I would like to thank Mike and Gore Lab members for their support in this project. I would like to thank RU student, Akriti, and undergraduate student from UCSD for helping with GC phenotyping and 3' RNA tissue collection. I would like to thank another RU student, Elise, for genotyping work, and Dr. Borgott from Algoma University for performing wax analysis. I would like to thank NSF for providing the funding for this project. Thank you all for listening, and I'm glad to take any question.